The lac operon is a genetic mechanism used by bacteria to decide whether they should either use glucose or lactose to make ATP. So why do you have to know about the lac operon? Well, honestly, this is something that has no clinical utility by itself, but is taught and tested because it's a classic example of gene regulation, and the same principles apply to tons of other things that are more relevant to medicine. Okay, so how does this work? Well, first, it's important to understand why the cell cares what substrate it's using in the first place, which comes down to energy efficiency. The cell would really prefer to use glucose, but it's okay to use lactose instead if no glucose is available. A key point here is that the cell needs to expend some energy to produce and maintain the enzymes and machinery required to metabolize lactose. So if glucose is available, then there's no need to synthesize these lactose metabolizing enzymes. That's where the lac operon comes in. It senses how much glucose and lactose are available and uses that information to decide whether or not to produce the lactose metabolizing enzymes. Okay, let's go through some examples. If both glucose and lactose are available, does the cell want to synthesize the enzymes that metabolize lactose? No, because remember, the cell prefers to use glucose. So if both sugars are available, it doesn't really care about the lactose and won't waste energy making the enzymes needed to metabolize it, since it's happy using glucose alone. There will be very low expression of the lac enzymes, and I'll explain why that's the case later. Let's do another one. What if there's no glucose but there is lactose? In this case, the cell wants to make these lactose metabolizing enzymes because only lactose is available, so this is what you're going to use to make ATP. Next, hopefully these are getting easier now. If you have glucose and no lactose, do you want to make the lac enzymes? No, because you have glucose and there's no lactose around to metabolize anyway. And lastly, if neither glucose nor lactose is available, what do you want to do? Well, again, there's no lactose available, so it'd be a waste of resources to make the enzymes needed to metabolize it. Alright, now let's go over the molecular interactions that allow the cell to make these decisions. This gray bar is DNA, and the genes needed to metabolize lactose are these: LAC-Z, LAC-Y, and LAC-A. And upstream from these genes is where we'll regulate whether or not we want to transcribe them into mRNA and then make the proteins that correspond to these genes. An important point here is that since this is a prokaryotic system, we can make polycistronic RNA. Do you remember what that means? Polycistronic mRNA has more than one gene on it. So in one continuous round of transcription, we make an mRNA molecule that has these 3 different genes on it. This does not happen in eukaryotes like us. Now let's walk through how we get transcription. When do we want to transcribe these genes? When there's no glucose and plenty of lactose. So, when glucose is unavailable, first, the enzyme adenylyl cyclase converts ATP into cyclic AMP. Cyclic AMP can then bind to an activator protein called catabolite activator protein, or CAP, causing it to bind near the promoter and help recruit RNA polymerase to transcribe the genes. But wait, we only want to transcribe these genes if we have lactose, right? So how do we make sure we don't transcribe them if there's no lactose? Well, we have this inhibitor protein, which is encoded elsewhere in the genome by the gene LAC-I, where the I stands for inhibitor. This protein binds to a region called the operator and prevents RNA polymerase from transcribing the LAC genes. However, if lactose is available, it binds to and inhibits this inhibitor, allowing transcription to proceed. So if we have lactose, we can make the proteins that correspond to these 3 genes, which are needed for lactose metabolism. Now what about glucose? Well, if glucose is present, we don't want to transcribe these genes. So the enzyme adenylyl cyclase has a binding site for glucose where glucose can shut it down. So the cap protein will never bind to the CAP site or recruit RNA polymerase to the promoter. Now this particular mechanism isn't 100% effective. As I mentioned earlier, if you have both lactose and glucose, the LAC-I protein won't bind to the operator, and CAP won't be activated. But you will still get a very low level of transcription of these genes. On the other hand, if you don't have lactose, you get pretty much zero transcription because the inhibitor protein is a lot more reliable.